Hi, welcome to this video. My name is James Cox, and I'm the Metabolomics Core Director here at the University of Utah. Today's video, I'll be going over who we are, where we're located, what we do, how we do it, and how to understand a data set that we send you. As of today, we have 5 staff members and 8 state-of-the-art instruments. Contact information for each one of us can be found at our website, chd.cores.utah.edu/metabolomics. We're located in the basement of the Emma Eccles Jones Medical Research Building, room A306. To get there, you enter the front door. Here, on the right-hand side, you'll see a door. It says stairwell or stairs, and you go down the stairs. And at the bottom of the stairs, you'll see a door. You go through the door, you go down the hall, and then you'll see the Metabolomics Proteomics core on the left-hand side. If you don't live in Utah, you can send packages to us. Just pack your samples on dry ice and then send them to us by UPS or FedEx to this address here. This is also listed on our website. Send us a tracking number for your package and we'll track it for you and alert you when it arrives. So what do we do with the Metabolomics core? Well, we do metabolomics, of course, lipidomics. We also do fluxomics and isotope tracing and mass spectrometry or just general metabolite quantitation. And this could be of peptides and small molecules. In general, for metabolomics and lipidomics, we work from a non-targeted discovery approach to a targeted quantitative validation approach. We use GCMS and LCMS for metabolomics, and for lipidomics, we mainly use LCMS. As mentioned before, we have 8 state-of-the-art instruments that work from the non-targeted discovery approach to the targeted quantitation. As I also said, that we usually are using quadrupole time-of-flight instruments. So we have several UPLC QTOFs here and here, and we also have an Agilent GC QTOF. Once we discover an altered metabolite found in your samples, we then try and validate that alteration using a targeted quantitative approach, using either triple quads for LC systems or a single quadrupole for a GCMS. So metabolomics, what is it? Well, if you're at this video and been to our website, you probably have a pretty good idea of what metabolomics is, but just to be clear, it's the study of the low molecular weight compounds that make up metabolism. And you have no doubt you've seen this type of pathway chart in biochemistry classes or other metabolomics presentations, and it makes it appear that there are literally hundreds to thousands of metabolites found within a cell and cellular system. But there are some serious caveats to metabolomics. One is not all metabolites are observed by mass spectrometry, and there's a lot of reasons for this. So they can be unstable, they can be found at low concentration, they can have low ionization efficiency, they can have poor extraction efficiency, or they're covalently bound to a protein, or they're stuck to a protein in some other way. And so another major caveat is really no single method is perfect for observing all metabolites in a single sample. And there's a lot of reasons for this. And a lot of it's because the charge states of metabolites are different. You can have a positively charged carnitine, but if you try to run that in LCMS in negative mode, you won't observe it. And also, there's a wide range of hydrophobicity and hydrophilicity of metabolites. And this requires us to use different chromatography methods. And so, to really get a full profile of the entire metabolome, multiple modalities are often used to do this. This brings us to, well, what do we choose? Metabolomics GCMS or metabolomics LCMS? I really kind of think of it like this. GCMS is really good at really small molecule analysis. This includes free fatty acids, amino acids, TCA, cycle intermediates, and sugars. And most of those sugars we can do single phosphorylated sugars and some diphosphate sugars. But where LCMS really shines is in bigger metabolites, such as redox metabolites, NAD, NADPH, glutathione. Also does really well on phosphorylated nucleotides and really nucleotides in general. So this brings us to the question, how do you pick a modality, GCMS or LCMS, for your project? Well, first, the best way to do that is to contact the core. Before you start a project, contact us to determine which analytical strategy is the best. Also, we have a whole other video, Sample Submission Facts, that describes how to prepare and submit samples to us. But basically, you go to our website, fill out the sample submission form and submission details form shown here on the right-hand side. Fill those out, email those to us, and then ship them to us or bring them to the core. Again, also see our whole other video, Sample Submission Facts, for more details. So next we'll go over the 5 steps that we perform in the core to prepare your samples and then to see new data. First, we prepare your samples by extracting the metabolites out of them, and then we put them on an instrument, and then we take the data that comes off the instrument and extract it, and then we prepare that data for you, and then we build a report so you can understand it. So the first step is sample preparation, and basically what we're doing is we're extracting the metabolites from your sample. It can be serum, it can be tissue, it can be fruit flies. But in general, what we're using is a cold acetonitrile, cold methanol addition to crash out your proteins and leave the metabolites in the supernatant. If it's a tissue, we'll often use bead meal tubes to break up that tissue, and then use centrifugation to remove the tissue and pellet it. And then once we have the pellet, we take the supernatant and put in autosampler vials. We also have a number of publications where you can find the details for these extraction methods shown here. So once your samples have been extracted, we then run them on a mass spectrometer. And there's a couple aspects of this that are important to understand. One is we use process blanks and quality control samples to determine how well these instruments have run and how good your sample data is. And so what a process blank is it's a sample that's undergone the same extraction steps as your samples, except instead of having tissue or serum or anything like that, it's actually just deionized water. What QC samples are is 5 or 10% portion of every one of your samples that we've extracted. We put them into a pool and then divide them up into individual samples. And we run those within the sample set themselves. And what we're doing here is the first several QCs are used to condition the column before we run further samples. And the third QC shown here, this is actually QC where we just use to identify if it's actually a good run or not going to be a good run. In other words, is a mass spectrometer performing where we want it to perform? A second aspect that's important is we do all your samples in a randomized order using a random number generator. And this is important. We don't want to put any kind of sample bias into the data set that will give you false positive results. And then we run random QCs every 4 to 6 samples shown here and here. And lastly, we run a very last process blank to see how much carryover chromatographic contamination has occurred throughout the sample run. I've already discussed the mass spectrometers, but this is the output that a mass spectrometer does, the chromatogram. Now these chromatograms have a lot of peaks shown here, but it's really almost extraordinarily difficult to determine if there's any real alteration in your samples just based on the chromatogram itself. So once we run your samples and we have a chromatogram, we need to convert that chromatogram into useful information. The way we do that is we process these samples through a mass hunter quant. And what that does is it identifies the metabolites based upon the fragmentation pattern shown here, and then quantifies the area under the curve of the peak that it corresponds to. This does this for a number of metabolites, all the metabolites that we tell it to do it for, and then what it does at the end of the day, it produces a .csv file with has area under the curve for each metabolite that we're interested in. So the next step we do is we take that raw data in that .csv format and have to make it into a format that we can next interrogate. And to do this, we had a postdoc in the core, Brad Naylor, write a script or a program for us called the reporterator that takes that raw .csv file and goes through a number of steps to produce a finalized data report. So this is the GUI for the Reporterator software, and basically we have a number of functions that we can do here, shown up here, and we just choose whichever one that we want to build a report. And I must tell you right now, a lot of these scripts that are used for %CV, LOES, normalization, compound renamer, we have taken from various freeware scripts. And one major place that we took a lot of this from was from metaboloanalyst.ca, which I'll show you in a moment. So the first step that we do is we do a %CV analysis, and what this is a % coefficient of variation for each metabolite, that QC cohort. So metabolites at a higher %CV of 30% are removed. The next step, in larger data sets to adjust for instrumentation drift, we will then do something called LOES. And so what LOES does is it goes through those equally spaced QC samples throughout the whole set and then looks to normalize for drift. Third step is we do normalization, and we do normalization for internal standards that we've added to each sample. And we will also do normalization to adjust for cell number differences, tissue weight differences, etc. And then we'll make a final report. So this is nice. It automatically produces a final Excel file for you to further interrogate, and actually builds a nice Word report. So let's look at the Excel file that is produced by the Reporterator. All the data preparation steps are included in separate tabs that were performed in the preparation of your data. But the most important tab here is the Metaboloanalyst input tab. You can click on that tab, copy and paste that, and save as a .csv file, and then go to metaboloanalyst.ca and do further processing of your data to do different statistics with it. We also generate a final report, and this final report describes your experimental goals that we've done, or what you wanted to have done. And then we have an experimental section that we actually take all the details that we did for the extraction process, the mass spectrometry process, and how we did the data analysis shown here. And we designed this so you can copy and paste that and put that right into a publication format. But the most important thing I want to stress here is please cite our grants. This is how we get more instruments, and we have 3 different grants shown here. S10 grants that have funded the instrumentation purchases in the core. Now, another thing this report has is if you want to do the statistical analysis on your own, we have a detailed step-by-step instructions that we've done here that you can mimic the data processing that you'll see further through this report. And also, we include a description of all the tests that we've used to determine alterations in your samples. So this is further down the report. What we do is we compare your samples and whatever comparisons you ask us to do in the Reporterator, or you can do this in Metaboloanalyst yourself. We make heat maps, and we also can do dendrograms, shown here on the right-hand side. And these make for really nice pictures, but they really don't tell us too much about the metabolites that are altered that change and allows for groupings. We also include principal component analysis for historical reasons. They are commonly found in metabolomics papers. The first plot on the left shows the first 5 principal components, and the second plot on the right shows just the first 2 principal components, and this is where most of the variation is going to be found between the 2 sample types. You can also use the loadings plot derived from this data to identify what metabolites contribute to this class separation, but this is really tedious and we don't do it. The next plots that we just show are partial least squares discriminant analysis. And this is really similar to principal component analysis, but it's a directed form of this. So basically, you're telling the software what samples belong to which groups, and then the software determines what is the most different metabolites between the two groups. The problem is it does not contain any statistically relevant information such as p-value and fold change. It just simply finds differences. But a useful aspect of PLSCA is it produces a VIP scores plot shown here on the right, and this gives you an idea of what metabolites are contributing the most to class separation. And so lastly, what we often send out is a volcano plot. Volcano plots is a workhorse graph when comparing two conditions and is my favorite visualization tool. What you're doing is you're prioritizing altered metabolites based upon their p-value and their fold change. So you can sort through this data table shown here on the right and determine the importance of each metabolite based upon these statistical parameters. And this is limiting the number of metabolites that you really need to consider when you're searching through metabolic pathways. And so for this data set shown here, we have a rather stringent alterations of two-fold change between the mutant and the wild type and p-value 0.01. But if you wanted, you can take that data in MetaboAnalyst format input file that we have, and you can change that in MetaboAnalyst.ca on your own. And lastly, thanks for your attention. If you have any questions, please feel free to contact us, and we are funded through a number of different mechanisms. Thanks.